Thanks very much. Good evening, everyone, and I'm delighted to be able to present ReNeuron to you again this evening. And apologies for those of you that may have already seen something similar to this or otherwise know the company. Hopefully it won't be too boring for you. And for those of you that don't know the company, hopefully I can enlighten you. So there's our disclaimer. I'll straight past that. So we are a pharmaceutical business, a biotech business developing stem cell therapies. So we're trying to harness the power of stem cells and convert those essentially into therapeutic products, targeting a range of diseases where conventional approaches either haven't been tried or haven't worked. And that's the real essence of advanced therapies, cell and gene therapies, is addressing major unmet medical needs. We are an AIM-quoted business. We're based in the UK. We have sites in the UK and in the US. We're currently worth about 55 million, of which half is represented by cash on the balance sheet, at least last reported cash. Critically, we're developing allogeneic stem cell therapies, and that means non-patient specific. So in other words, we're trying to develop standardized cell products that any patient with the diseases we're targeting can have administered to them. The alternative approach is autologous, where you're using the patient's own stem cells, and that clearly does have merit in certain settings. But for a business like us, and certainly with the diseases we're looking to treat, a non-patient specific approach, we believe, offers the best approach to ultimately a commercial product and one that's going to deliver clinical benefit. We've got 2 clinical stage therapeutic candidates in the pipeline at the moment, and I'll take you through those in turn. And we believe we have some very significant clinical data points coming up over the next couple of years, and that starts very soon, actually. And really, that's the essence of how value is generated in a business like ours. We don't generate revenues. We raise money either on the stock market or through other means, non-dilutive financings. And then we try and spend that money to generate clinical data with our therapeutic programs. And if those data are positive, and that's not always the case, there's risk here, but if those data are positive, then there's a very significant valuation opportunity there, because that's when these programs become valuable to potential partners or acquirers. And we follow a pretty traditional biotech model. We hope in due course that these programs will be licensed or acquired by the larger companies that have the clout to take them through to market and into their marketing distribution systems. So the 2 assets that we have are listed here. The first of them is called HRPC. That stands for human retinal progenitor cells, and don't worry too much about the terminology. The second asset is CTX, and that's a neural progenitor cell line. So in both cases, these are stem cells which have the ability to differentiate into the types of cells that are appropriate to the organ of origin, from where we originally derived those stem cells from. So in the case of the HRPCs, they are essentially retinal cells. So we're targeting those at retinal degenerative diseases, the first target being retinitis pigmentosa, and I'll come back to that disease and what it's about. And in the case of the neural cells, we're targeting stroke disability. So again, a brain condition. So we're kind of believers in using the cell type that's relevant to the organ that we're trying to treat here. In the case of both of these assets, we've partnered them for China with Fosun Pharma. Obviously, some of you will be aware of Fosun and their exploits with Thomas Cook and Wolverhampton Wanderers and other investments in the West. But in the East and in China, they are a major healthcare player. They're probably the top second or third pharmaceutical company in China. They have massive clout over there. So we couldn't have hoped for a better partner to license these technologies really quite early in their development for that market exclusively. So that sort of seals up China for us and obviously leaves the way open to do other partnering deals in due course elsewhere in Asia Pacific and in the West, of course. We also have an emerging exosome-based platform. If there's time, if I don't run out of time, and I usually do actually, but if there is time at the end, I'll take you through that very briefly. But it is earlier stage. It does have some high potential. It's very research-focused at the moment, but it's deriving exosomes from one of our existing products, from our CTX cells. and it's something that we're pretty excited about. So the pipeline is listed here. Every biotech company has its therapeutic pipeline, and this is ours. And as you can see, our top, or our 2 cell therapy candidates are both in phase 2 clinical development. So they're midway through the clinical development pathway. And as you can see, in the case of HRPCs, we're looking to generate, in fact, we are in the process of rolling out some interesting phase 2A data this month, and there'll be a conference where more data will be presented this weekend, actually, at a conference in San Francisco. That's looking very good, and I'll come to that in a second. With our CTX cells, these are targeting stroke disability, as I said, and that's in the middle of actually a much longer, more expensive and complex phase 2B study, again in the US. All of our clinical work at the moment is taking place in the US. It's obviously a major potential market for us, and that's why we're doing those clinical trials there. The readout in that phase 2B study is sometime in the first half of calendar 21. So looking at the HRPC program first in a little bit more detail. So as I mentioned, HRPCs are allogeneic cells that have the ability to differentiate into, in this case, the types of cells that are lost typically in retinal degenerative diseases like retinitis pigmentosa. So in other words, photoreceptors that give us sight. Rods and cones, in other words, that reside in the retina. And we've shown preclinically that our HRPCs can differentiate into those lost photoreceptor types and engraft into the back of the eye, basically. And what we believe they're doing is thereby replacing some of the photoreceptors that are lost in these diseases and also providing support, trophic support, to photoreceptors that still survive. And that's the essence of cell therapy, either to replace lost cells or to provide signaling mechanisms, or through signaling mechanisms, to provide a trophic mechanism that promotes repair and protects cells that survive. We're targeting retinal degenerative diseases, obviously, for that reason. And very importantly, not only as a business do we need to generate unequivocal clinical data, hopefully positive, but also we need to show that we have the means to manufacture or expand these cells in a GMP, a good manufacturing practice environment, at scale, at a cost of goods that's going to be relevant and of interest to a potential partner. And that's a box that we've certainly ticked with this particular candidate. And it's also the case with our CTX cells as well. And the business was very much founded on the essence of an allogeneic approach that means you can actually generate ultimately cells at a cost of goods that is manageable. And that's going to be very important when it comes to getting these products priced, adopted and reimbursed in Western healthcare systems. So retinitis pigmentosa, the first target, as I mentioned, is an inherited degenerative disease of the back of the eye. It usually manifests, as you see in the images on the right there, in a loss of peripheral vision initially, but ultimately it leads to blindness. There are no existing treatments bar one, Luxturna, which addresses one of the many gene defects associated with this inherited condition. So the vast majority of these patients still have nothing available to them, unfortunately. It's called an orphan drug target because it's relatively rare. It's actually quite a large disease among orphan diseases. And for that reason, we have orphan drug designation for it, both here in Europe and also in the US, and fast track designation for the program by the FDA, the US regulator. And that's important because orphan drugs typically can command a swifter route to market. And when they get there, they can command a degree of market exclusivity for a period of years as well. So orphan drugs tend to be of great interest to potential partners and acquirers for that very reason. I'm not going to take you through the preclinical data. Suffice to say that we've generated and published a raft of preclinical data that show the putative benefits of HRPCs in terms of their ability to exert beneficial outcomes in models of these retinal degenerative diseases, mostly measured by visual acuity, the ability of an animal to detect light, to sense movement, pre and post treatment. So that's what got us into the clinic, and this is just a slide, actually, again, I won't go through it, that just sort of demonstrates that among the various models that we used to demonstrate to the FDA that this was ready for the clinic, we could show a durability of those positive responses over time, and that's also important. A transitory effect is okay, but clearly it's suboptimal. to something that actually has lasting benefit at the right level. So that has led us into the clinic, and we're currently in the middle of a combined phase 1/2a study in the US, run at 2 centres under 2 very leading and prominent key opinion leaders, Jason Commander and Praveen Dougal, in those 2 sites in Boston and Phoenix. The phase 1 part of this study in 12 patients we've completed. It was a dose escalation study to show safety at an increasing dose of HRPC cells. And we've moved into phase 2a using the highest dose that was safe in phase 1. It was the highest dose used actually, which is 1 million cells injected straight into the back of the eye using subretinal injection. In the phase 2a, we're treating up to 12 further patients. We've treated 10 so far, as you'll see in a second. Primary endpoint is still safety, but we're also monitoring for visual acuity and other putative efficacy measures in those phase 2a patients as well. And what we've seen so far, as I mentioned earlier, is pretty encouraging. The image here is an ETDRS chart, and this is it's not, as you can see, it's not really the same as a Snellen chart that you would typically read down in an optician's, but it's a similar principle. It's somewhat esoteric, or rather more esoteric than a Snellen chart. But it is a fairly classic measure of visual acuity. It's a reasonable biomarker of visual acuity in clinical studies of this nature. It's not a perfect one, but it's one that's perfectly validated and accepted by the regulators and the field in general. And what we've seen so far, and the data is still maturing, and as I mentioned earlier, some of the more updated data will be presented at a conference in San Francisco this weekend. But in the patients that we've treated so far, at the follow-up points that they have reached in turn, we are in general seeing a very meaningful improvement in visual acuity in these patients, which is being sustained. Okay, so what we do need to do is let these data mature further, but if we see what we're seeing so far and these visual acuity improvements in terms of numbers of letters that the patient's able to read on this chart pre and post treatment, if those are maintained, then most definitely we'll have the ability to move forward into the next stage. And that is probably going to be a much larger pivotal study in a much larger group of RP patients. And our next next bit of the plan is to talk to regulators here in Europe and in the US to determine what that protocol might look like and when we might be able to start it. So we have very, very high hopes for this program, and we're delighted to have leading KOLs behind it. And it really is now a race to data. And as I keep on saying, that's really where we can drive value in this business, by generation of data along the lines of what we've already been generating so far this year. And I mean, our share price has certainly had its ups and downs over the year, but off the back of the early data in this study, it's more than tripled so far this year. But I've been in the business long enough to know that sometimes share price increases don't hold, and they can go up and down actually when you least expect them to. But generally, we're very pleased by the market reaction to the data as this has matured over the course of this year. So in the time I've got available, I'll just take you through the other program, which is really a much older program. It was the neural cells that we initially started working with when the company was set up. Perhaps the name of the company kind of gives it away. But it's the same philosophy. CTX is an allogeneic or non-patient specific approach to targeting damage in an organ where stem cells may play a role. In this case, it's damage associated with stroke, ischemic stroke, a blockage of blood flow to the brain, which is by far and away the largest type of stroke, the other one being hemorrhagic, which essentially is a bleed in the brain. And we've shown preclinically again that these cells have an excellent safety profile and have the ability to restore motor function in models of stroke disability preclinically. And again, we have the ability to manufacture this and cryopreserve it so it can be shipped and stored and used when required in a way that's akin to a more standard biopharmaceutical. So it's trying to convert stem cell science into living cell products that are easy to use and administer and ship. In terms of how these cells work, in common with other cell types, there's plenty of literature around this now. It's known that certain cell types, including neural progenitors, exert their beneficial effects in a number of different ways. We know that these cells stimulate blood flow in the brain, so they're highly proangiogenic. They stimulate neurogenesis through cell signaling, and it's probably the exosomes actually that mediate that. And they have the ability to modulate the immune system and stimulate synaptic connectivity as well in the brain. These are putative mechanisms. There's a lot that we still don't know about cells, our cells, and also the field in general. But we know enough empirically to know that these cells do have a beneficial effect, enough to get this program again into the clinic. The target is stroke disability. It's a massive unmet medical need and a huge drag on health and care systems around the developed world. And this is a chart that I usually put up actually, which demonstrates why this is important to us. And modified Rankin is a very, very good measure of measuring disability or dependence in patients who are disabled by their stroke. It's a 5-point measure. And the higher up you are on this measure, the more dependent you are, as you might be able to see on the charts on the right hand side there. And the bar graph is taken from a study that was published in Sweden a couple of years ago in over 40,000 disabled patients. And the axes essentially, what it tells you is the pharmacoeconomic benefit of reducing a patient's level of dependence. By knocking them down 1 or maybe 2 levels on this measure, you garner very substantial annual healthcare savings, and that's actually the y-axis. And sorry, it must be very difficult to see, so apologies for that. But the important thing about this slide is that modified Rankin is where we saw interesting data at phase 2A, and it is the primary endpoint in our ongoing phase 2B study in the US. Our phase 2A study, which was run in the UK actually, in 23 stroke patients, single-arm study, so we were monitoring here the patients against a baseline level of disability after their stroke. We were making sure that those patients had a stable level of disability before they got the treatment. And if you do survive your stroke, generally within 3 months, if you suffer a motor deficit, it will stabilise within that time frame, and most certainly by 6 months. Many stroke patients will go on and make further cognitive recoveries, but their motor deficits, this unilateral inability to move down one side of the body, will have stabilised within that time frame. And that's important because what we want to do is eliminate the effect of spontaneous recovery as a confounding factor in our current study. Anyway, against the baseline, what we saw in the phase 2A was a meaningful response level on modified Rankin, enough to justify a phase 2B study and actually comparable to treatments that have already been approved for acute stroke, the most notable one being Alteplase, which is a Genentech drug. In fact, it's the only approved treatments for stroke patients in their acute phase. Importantly, if we eliminate patients that had no ability to move their upper arm going into the study, the response rate's even higher. And intuitively, that's what we expected, because we suspected that if a patient couldn't really move the arm at all going into the study, it was very unlikely that they were going to be a responder. And the data seems to bear that out, albeit these patient numbers are obviously pretty small. The phase 3 study is a much larger one. As you can see, it's in over 100 patients, randomized either to the treatment, which is a neurosurgical implantation of cells, or to placebo sham surgery. We're treating 6 months post-stroke for the reasons I mentioned earlier. The primary endpoint is modified Rankin 3 or 4, and again, if we can drop patients down 1 measure from 3 to 2 or 4 to 3, that's clinically significant for the patient and in terms of hitting the primary endpoint in this study. We're also monitoring on a number of secondary endpoints here, and again, without going through them, they are all pretty conventional in clinical studies run in disability, basically. There's a large number of sites in the US involved in this study, and it will read out in the first half of calendar 21, all being well, as I mentioned earlier. I've got 2 minutes left, so just a very, very quick word on exosomes. And we've learnt a lot about how our CTX cells might be exerting their beneficial effects. And exosomes actually is an emerging field over the last few years, both in terms of the use of exosomes as a delivery vehicle for delivering advanced drugs, or as a therapeutic agent in its own right. And we're exploring both angles here, but our primary focus actually is on delivering third-party advanced drugs using our exosomes essentially hollowed out. And we believe that exosomes as a delivery vector offer advantages over the best standard at the moment, say for delivering a gene therapy, which is an AAV vector, and over and above that, we believe that our particular exosomes have certain advantages over others that we see. Most notably, they are derived from a candidate that is already a clinical agent, i.e. the CTX cells. Anyway, without going into the science in any more details, I'll probably trip myself up, not being a scientist by training. We have high hopes for that program, and the way that we're developing our exosomes is collaboratively. We're not, in the case of our cell-based programs, we're taking those forward to market or close to market under our own steam. In the case of exosomes, we're quite happy to work collaboratively, as I said, to deliver a third party's drugs, and we'll share the spoils if we have success with those endeavours. So just to wrap up, we're a global leader in cell-based therapies. We're one of the pioneers actually in the field. It's an allogeneic approach which is patented, scalable and cost effective. We're targeting unmet medical needs, which is the essence of cell therapy success as far as we're concerned. We have some significant clinical milestones ahead of us before too long. And in the near to medium term, we also have the ability to do partnering deals to generate more value and to bring more cash into the business. And our deal with Fosun this year was the first such example of that. And with that, I think I am out of time. So thank you very much.
Michael, thank you very much. Thank you. Okay, as I mentioned, if you have a question, please raise your hand and wait for a colleague or me to come over the microphone. So, gentleman at the back here, please, first of all.
Hi, I'm quite interested in how long it takes for people to recognise they have a problem to get through to the point where treatment can begin. Does that have an effect, or do you need to get people immediately? Problems occurs?
No, actually, I mean, in both cases, these are... sorry, can you hear me okay?
Yeah.
These are treatments that are essentially addressing chronic conditions or degenerative conditions. So they don't, although stroke is of course a sudden insult to the brain, we are looking at the downstream consequences of stroke. So in that sense, these patients, essentially we're taking them out of the community, they're already back at home and have survived but have a disability. And in the case of retinitis pigmentosa, of course, it's a disease that progresses over many years, in fact decades in some cases. So in that sense, it's not an issue in terms of, you know, we have to get a patient into hospital or, you know, or into the surgery or whatever to get these cells or to get the treatment before it's too late. It's not, there's not an imperative in that sense.
Yeah, because I'm sort of thinking about how difficult it is to get through the GP system to actually be diagnosed.
Yes.
Does that have an effect on the treatment you can offer?
Yeah, in a clinical setting, actually, I mean, we've learned a lot about how stroke patients typically are triaged in various jurisdictions, including here in the UK and in the US. And it does vary. The standard of stroke care is pretty spotty, actually, depending on where you are, where you live within a country or between countries. But generally, there are systems available there for stroke patients in order to be able to identify them and get them what they need. The problem with them, of course, is that at the moment they have nothing available to them other than sporadic rehabilitation or physiotherapy if it's available, you know, where they live. So it does vary.
Okay, thank you.
Other question? I'll bring the microphone over.
Hello, I've got several questions. First of all, could you just give some kind of indication of, I know it's variable, how long it is to approval, and the cash flow that you have got to sustain before you get to the point where you're beginning to get some money back. Also, how other alternative companies in the field doing similar or the same thing? And are you liaising with local stroke centres in Manchester?
Okay, so take them in reverse order. No, we're not liaising with local stroke centres at the current time in the UK because, as I mentioned, all of our current clinical work is taking place in the US. So we have to sort of make choices about where we do clinical work. We did our first 2 studies in the UK. At the moment, because we're doing a much, much larger study, we need to have access to a critical mass of centres, both recruitment and surgical centres, and the US actually is by far and away the the most amenable territory to do that, at least to get a trial of this complexity done in reasonable time. But that's not to say that, you know, we've abandoned our home territory, if you like. I mean, clearly, as we make more progress, we see this as a treatment that should be available for patients in any territory that has the ability to treat them or administer the treatment and get it adopted. In terms of the cash situation, we are, I mean, basically, I think we have got a pretty long track record of raising money, both through equity finance, through grants, and more recently through business development activity to get us to where we need to be. You know, have we got enough cash to get both of these products through to market? No. Am I confident that we can get these products through either to market or to the position where they're out-licensed? Yes. Will we need a huge amount of new cash to do that? No. Part of the reason for licensing is, of course, you know, ultimately we'd love to be able to keep these programs to ourselves, take them all the way through to market and market them. But that's an unrealistic proposition. So we have to make a judgment as to when we've delivered enough value in the programs to make them valuable to a partner and to deliver a return to our shareholders. And if the data in either one of these programs is positive at phase 2B, then that value will be there, undoubtedly. And part of the reason for that speaks to your other question, which is we are not overly encumbered here by competitive threat. Quite the opposite. And that's really a function of what we're doing. So much of what we've done as a company over the last 20 years has just not been done before or by anyone else, either before or since, actually. So there are a number of other players that are looking at cell and gene therapies around the world in a raft of different indications. In fact, advanced therapy, as it's known, is a rising field at the moment and has definitely been cottoned onto by large pharma. And that's evidenced in some of the very high-value deals that you've probably seen with cell and gene therapy companies, CAR-T and the like over recent years. So, you know, our mission, and I guess what we're paid to do, is to deliver that clinical data, hopefully positive data, and then seek to derive value through commercial activity, most notably business development activity, when we're ready. And you know, we're not far off that. We have a phase 2B in progress with stroke and a phase 1/2A in an orphan indication, which offers a much swifter route through to proof of concept.
I think that's perfect, Michael. Thank you very much indeed. Thank you very much.
Thank you.